Hello, today we are going to discuss DNA replication in prokaryotes, which occurs in the cell cytoplasm. DNA replication is semi-conservative, meaning that the double helix is split, and each strand is used as a template for new DNA to bind to. We will use the theta replication model to demonstrate DNA replication. To start, the origin of replication is a sequence of nucleotides where replication begins, and once an initiator protein binds to this region, it causes the DNA to unwind and split. Helicase is an enzyme that can attach once the DNA is split and continues to unwind and split the double helix into two individual strands. When DNA strands are split, single-stranded binding proteins come along and attach to the individual strands, stabilizing them and preventing the strands from rebinding. This is important to ensure the double helix stays split and replication can occur. Another important enzyme is gyrase, which attaches to the DNA double helix ahead of the replication fork and nicks DNA to relieve tension due to supercoiling. Supercoiling is when the DNA twists around itself due to over or undercoiling. This can create issues for replication, so gyrase is key in preventing that. Looking at the two strands of DNA, we can see that DNA replication can occur bidirectionally from the origin of replication. Here, we have continuous replication, which is uninterrupted and drives into the replication fork. This strand of new DNA is the leading strand, as it is replicated in the 3' to 5' direction and follows the replication fork. On this side, we have discontinuous replication, which drives away from the replication fork and is interrupted because synthesis can only occur as the replication fork opens more. This strand of new DNA is the lagging strand, as it goes away from the replication fork and is broken up into different pieces. These individual fragments are called Okazaki fragments and will form one continuous strand of DNA with the help of a few enzymes. On the bottom strand, we can see the opposite is true, with the leading strand on the right side and the lagging strand on the left side. DNA polymerase III attaches nucleotides to the growing DNA strand, but needs a free 3' hydroxyl group to bind to. The leading strand will always have a free 3' OH group, so DNA pol III can add nucleotides continuously. As the replication fork opens wider, there needs to be the creation of a free 3' OH group on the lagging strand for replication to occur. The enzyme primase places an RNA primer that allows for DNA pol III to add to it. DNA pol III then adds nucleotides until the next Okazaki fragment. DNA polymerase I is an exonuclease, meaning it removes RNA primer placed by primase at the end of nucleotide strands with DNA. However, it cannot create phosphodiester bonds ahead of the polymerase, thus there is a hole in the DNA called a nick. An enzyme called ligase binds to the DNA and creates the sugar phosphate or phosphodiester bond between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of another nucleotide, completing the newly synthesized strand of DNA. In the theta model, DNA replication is bidirectional and results in two circular DNA molecules. If DNA pol I was not able to accurately replace the RNA primer with DNA, then there will be errors in replication. There will be RNA bases present instead of DNA, which would cause the transcribed RNA to be incorrect. This would impact final protein or enzyme production and could prevent that from happening due to inaccurate code. This would harm the organism as they could not produce the necessary proteins or enzymes. Here's a list of my references. Thanks for watching.